So everybody, welcome to our webinar today. As the title shows, the webinar is about high dimensional analysis and visualization of flow cytometry data. In contrast to most of our webinars, this is not going to be a step-by-step introduction to features in FCS Express. If you were looking for like a tutorial how to use FCS Express or how to use high dimensional visualizations, this is not the webinar for you. This webinar is really an educational webinar designed to go over some of the background concepts that you need to understand when using these tools and why these tools work and why they're necessary and what they do. And that way, you know, there are more and more tools coming out and you'll be better suited to understand which ones to use and why they're working and why they're not working. So that's a little bit about what this is about today. As Raphael said, you know, during the webinar, please feel free to type in any questions during the webinar. Raphael will answer them back in the question and answer box, or if it's something that I can sneak the answer into the webinar, if I feel it's a really good general purpose question, I'll try to sneak it into the context of the presentation. Okay. So this is a slide that we got from one of our customers that I think really accurately, this is a question we got or a comment that we got from one of our customers that really accurately represents, I think, some of the problems that people are going through. And I'll just read out bits and pieces. We were happy living with Diva and Prism until all these plots with Spade, Tisney, and heat maps came out. Since 2013, I'm struggling to find version of R as a way to use them. It has not been easy. I'm not a coder. The appetite for Tisney and Spade has grown significantly. The scatter plots of Prism are outdated. Grants and manuscripts all want these new plots. Basically, is then complaining that Spade, even though it was announced with a lot of fanfare already within a year, stopped being that popular. I spoke to some active users and they suggested Citrus. Initially, it was running from MATLAB, which is another thing to learn. Last summer, Citrus came to RStudio, but then I was told that Acedes 0.5 is something people like. Believe me, we old school flow biologists are spending countless hours to learn R and get the needle moving. It is an existential threat for us. This experience has not been easy. We have 300 subjects with 7 flow panels and 12 stimulation conditions. Imagine my frustration. And so I really like this you know, example because, you know, from a personal point of view, it's great that somebody acknowledges that flow cytometry data analysis can be an existential threat, which is really cool if you make flow cytometry software. But the other thing is that he highlights on really a lot of issues, you know. There's a lot of new algorithms that come out, Spade, Citrus, Acedes, FlowSom. How is a normal person supposed to be able to evaluate and understand when these work? You know, people became biologists because they like biology and they're good at biology. They're not necessarily mathematicians. So, you know, what are you supposed to do when 15 different people are all telling you 15 different algorithms are the best, right? It's a complicated, it's difficult. The other issue is even if you do decide that you trust one of those people and you want to implement one of those algorithms or a bunch of different algorithms, how do you do it? Well, most people went in biology because they didn't want to become computer programmers. But a lot of the sort of wisdom that's being put out there is like, oh, just learn R. It's so easy. It's so friendly. It's no problem. Anyone with half a brain can do it. And it's simply not true. I mean, R is a computer programming language, and there are people who like and enjoy and are good at computer programming, and there are people who don't want to do computer programming. And if you don't want to do computer programming, you probably didn't become a computer programmer, and maybe you went to become a biologist because it's much more interesting to you. And so telling a whole class of people that all they have to learn to do to analyze their biology data is become a computer programmer is, I think, a challenge that people have faced, and they're kind of rightly so. I don't want to be a computer programmer. So, you know, there's all this has come upon the flow community very rapidly. The explosion of colors in the instruments that they can acquire on a single cell has just grown tremendously in a very short period. And all of these techniques have been dumped on people with very little guidance about what it all means. And so that's what I'm trying to provide today, is some guidance about why and how and what is going on. And again, the problem is, you know, as sort of, I'm sure many of you seen this slide many times, you know, as the number of colors per cell, or the number of parameters or fluorochromes or epitopes, whatever you want to call it, per cell that's measured increases, there's just a lot more two-dimensional plots that you need to look at. Now, this is really, I think, an overstatement. I mean, nobody really needs to look at the viability tube against the viability channel against every single other color in their tubes, right? So, you know, it doesn't really increase this much when you add new colors, or new colors, but it certainly does increase, and it certainly gets harder and harder as you add more and more colors. These 2D plots just become more difficult to look at. And so people are trying to propose solutions for this problem, right? And the first solution is just draw more plots. I mean, that's the, you know, sort of naive thing to do. And in fact, so this is the one that's immediately, if I was trying to sell you my new algorithm, this is the one I'd immediately start making fun of and say how old school it is and how time consuming it is and how boring and how non-reproducible and you know, all those things that people say about these plots and these gates, you know, but actually at the end of the day, all the fancy graphs, you do have to end up going back to these two-dimensional plots to understand what these graphs are telling you. And we'll get into that a little bit later. So, you know, looking at the two-dimensional plots are still the only plots that are really biologically meaningful and really biologically interpretable. And so they're still really, really important, and you still really have to look at a lot of them. Okay, so I'm certainly not going to, you know, say that these should be disregarded or that they're not useful. They're absolutely critical. They allow you to use your own biological knowledge and your intuition, and as well, again, they're the only ones that show data in a biologically meaningful way. But there are other tools that might be able to help as well. So the second solution is to do really good complex math in multi-dimensional space and get an answer. So this is like a, you know, a machine learning type approach, or neural network type approach is common, where you try to train the system. You don't really understand what's going on inside the system, but you train it with sort of known results, and then you give it your experimental data, and it'll give you an answer, and you say that's the answer, and you don't know how it got there and what it used and how it arrived at that conclusion, but you just trust the answer it gave. So that's one way. That's not that common in Flow really at the moment, and so we're not going to talk about that anymore. And the way that is becoming common is just try to find novel ways of visualizing all this multi-parameter data on a single plot. Okay, and then when you get this plot, you have to figure out how to interpret this plot and what it means. All right. So in order to sort of go on, I just need to introduce a concept first, and that's the concept of binning. And people don't appreciate that all of their flow data is actually binned. Okay, so you know, the signals you get in the data you get in your FCS file, they're not the raw analog data off of the photomultiplier tubes. Okay, that even the virtue of getting it onto the computer means that that analog signal is put in a bin, it gets assigned a channel value, and there's no fractional channel values. So, you know, on a common, maybe the lower end machines these days, there's 262,000 approximately channels that it'll get assigned to. There's more on some other instruments, but it does get assigned to a particular bin. And then what's important is that your data that you're plotting is actually, your plots that you see are actually binned at a much lower resolution, okay, even than the 262,000 channels. And a bin is just really easy. You take your data and you make generally equally sized slices, and you just count the number of events that fell in every slice. So if we look at here, this is if we took our data and put it into 10 bins, which is what I did over here. This is going to be the count of events in your bins. And this is the histogram that you're going to get out of it. And people don't appreciate a lot of times that a histogram, a flow cytometry histogram, is actually just a bar plot, just like the one over here. It's just that we only draw the tops of the bars. And it looks like a continuous line because there's actually usually 256 or 1024 bins. So the distance between the tippy tops of every bin is very close, and it makes it look like it's a continuous line, but it's not. Your general flow cytometry histogram is actually a binned representation of your data, binned at a much lower level than, or much lower resolution than the actual raw data. Okay. And to show you that the binning is really very important, I hope this animation comes through. You need to match the binning to your data. All right, this is a first sort of arbitrary choice that you need to make. If you have if you bin your data into too many bins, you can't actually see the structure of your data. And if you bin it into too few bins, again, you can't see the structure of your data either. And here's a little animation that I hope it comes across in the GoToMeeting, where as the, oh, the animation's not playing on my screen. I am not sure why. So basically what the animation would show, this is too high resolution and you can't see any structure to your data. And as you lower the resolution, you actually see two very clear peaks. And then when you lower the resolution even further, the two peaks merge together and you only see one peak. So you need to match your data, your resolution, to the underlying data that you have. Okay, and the lowest number of bins I showed here where you really can't see anything in your data is 10 bins. And in fact, I'll show here the histogram looked like this after you did 10 bins. Okay, you didn't see any structure in your data at all. And in 256 bins, you saw some peaks in your data. Okay, and so what is the problem with high dimensional data analysis? Why is it so challenging? And it's challenging because of the so-called curse of dimensionality. Okay, and that's a really easy concept to understand. So if I have 3 possible bins in a single dimension, I'm binning my data into one of 3 possible bins. You can see that in 3 dimensions, in one dimension, I just have 3 possible bins. But in 2 dimensions, I already have 9 possible bins. In 3 dimensions, I have 27 possible bins. And by the time I go up to 12 dimensions, I have just an incredible number of bins to deal with. A huge number of bins are going on. And if we take something that's a little more useful, if we start here at the bottom, and we can see that if I take a bin resolution of only 10 bins per dimension, and we can see that 10 bins is not enough for flow data. It'll never be enough. But let's just take this really low number of bins. You can see by 10 dimensions, by 12 dimensions, so I have 12 colors, of which that's not even a high number. By 12 colors, I already have 10 trillion possible bins that the data can go into. Okay, and if we take a number of bins that's actually reasonable for flow data, 256 bins per dimension, you see that by 12 colors, I've got almost 8 times 10 to the 28 possible bins that my data can go into. Okay, and so you can imagine trying to search through and deal with this many bins just on 12 parameter data. If you go up to 20 parameter data, I mean, I couldn't even fit the numbers on the slide, is just mathematically challenging. And what you can imagine is if we even take the simple situation, the non-useful situation where I have only 10 trillion bins, well, if I have 1 million events in 10 trillion bins, you can see the data is going to become very sparse, right? I'm going to have a lot of empty bins because I'm only filling up a tiny, tiny fraction of my 10 trillion possible bins. And there's some other mathematical problems with having so many bins that I won't go into that much. So, you know, the key point to take away is that the number of possible bins that your data can go into, a cell can go into, explodes exponentially and becomes very difficult to work with computationally. Okay? And so what we need to do is we need to simplify this data. All right, and so how do we simplify this data? And so we need to reduce the amount of data in some way. We have too much data. We have too many bins, and also often we have too many cells occupying too many bins. And so whenever you reduce the amount of data, you're throwing away information, right? There's no way to get around that. If you have 1 million cells by 32 colors, and you say, you know what, I'm going to disregard some of those colors, and I'm going to disregard some of those cells, then by virtue of that, you are throwing away information in order to make the calculations and the understanding simpler. And the key to what we want to do is throw away information while preserving, or even better yet, highlighting information that we care about. So the thought or the concept is that not all the information present in the data is important for us at this moment. So let's throw away some information, and if we can possibly do it in a smart way, it might enrich the information. The remaining information might be enriched for what we are actually interested in. Okay, and that's what we want to do. And so how do we do that? Well, we can do that by throwing away individual cells. You know, if I've got 1 million cells, then that's a lot of data to deal with. Is there a way to distill that down to, you know, maybe 2,000 cells and work and analyze those 2,000 cells? Or I can throw away that empty space. You know, like I said, I have 10 trillion bins. Well, if I can throw away the bins that I'm not using and only focus on the ones I am using, maybe that can help me. And the other thing is maybe I can throw away unimportant features of the cell. Okay, so what is unimportant? Well, the idea is that I've got a 32 channel experiment with 32 colors or 20 colors or whatever it is. Maybe for the biological question I'm answering, a particular channel is not really that important, and I don't need to consider it in my mathematical computations. And so if there's a way we can determine the importance of the particular channels and throw away the unimportant channels, then maybe we can speed up our computations and do something with all that data. Okay, so one of the first ways that we lower the number of data points is by clustering data. Okay, so now we see sort of the advantage of clustering is that when we assign individual cells to groups or to clusters, and then just analyze the clusters from then on. Okay, so instead of having 10 million events that I'm dealing with, it turns out I only have 5 groups of cells here, and I'm going to then analyze the 5 groups later on. Okay, now clustering data we're going to talk about later has challenges and how do you do that, but the concept is, you know, reduce the number of data points I'm working with. I'm not working with 1 million data points anymore. From now on, I'm working with 5. Okay, and then you can look at statistics about those individual clusters. Okay. Some of the other approaches that I talked about just before, reduce the number of parameters. That is an approach taken by PCA. The classic example of this is PCA, although there's many others. And the idea is that PCA creates 2 new parameters, or as many new ones as you want, but usually people deal with the first 2 principal components. PCA stands for Principal Component Analysis. So here there's a plot of the 2 principal components. So I create 2 new parameters, but those 2 new parameters are comprised of a combination of all the other parameters. The only thing is all the other parameters are weighted in some way differently, so they contribute differently to PC1 and to PC2. And I won't go into how they're weighted and what it's trying to do, but the point is the idea is that we decide how important each dimension of the data is, and then use that information to select a subset of parameters or combine those parameters to create a new smaller set of parameters that we're going to work with. And in this little combination, you can see that if I combine all of the data from my, 6 colors, I can actually see multiple clusters here on a single graph. And those clusters are comprised of a combination of all the parameters together. So one would argue that there's actually 3 different cell types here potentially that are pulling out. Whereas when I look at all of this data here, it's very hard to see those cell types without further gating or doing anything further analysis. Okay, so that's what PCA tries to do, is create a smaller number of parameters. And somehow those parameters are, these new parameters are combinations of the existing parameters. And the last approach is to throw away the empty space. Those are the so-called manifold learning algorithms, of which the trendy one these days is t-SNE. And essentially what those do is those try, and we won't be focusing on this, I'll probably do another talk on t-SNE later, but it tries to focus in on the areas that have data in your space and represent just the areas with data in some sort of meaningful way. Okay? And again, if anybody has any questions, please feel free to, ask them in the question and answer box. Okay, so there's some important concepts that we need to know. I'm not going to talk about variance stabilization in this talk because I want to get through a little more, but it is really critical to talk about distance. Okay, what is distance? So, there are many ways to measure distance. People don't appreciate that in their day-to-day life. You think things are distance, it's like, oh, it's 2 miles away, it's 5 miles away, right? So, if our little scientist wants to go get some cheap sheath, you would say that the distance between the 2 places are 10 miles away and 1 mile away. You definitely say the 1 mile away is closer. It's a shorter distance. If I went along route B, I would go better. But actually, if we change it, and we say that route A, has a lot of traffic, and route B, you can go on a race car, you'd actually get much faster going on route B, right? So the idea of how far away something is a question that needs context. You can't just implicitly assume the distance metric that people care about, right? And a lot of these is reflected mathematically, is that there's very, very, very many different ways to measure the distance between 2 things. We're most familiar with the Euclidean distance, okay? So here, the Euclidean distance, the straight line between 2 points, all right? And this is the equation for it. But there's another common distance metric called the Manhattan distance. And the Manhattan distance is called the L1 norm or taxi cab distance or many other distances. And in the Manhattan distance, you can only go in 1 dimension at a time. Okay? And so one would say, well, which here is the true distance between those 2 points? And the answer is that there is no true distance. I mean, if you say that the Euclidean distance is the true distance, I can, that is the true distance. The other ones are just other distances. Well, I would argue that if I had a giant apartment complex here that was blocking the way, this Euclidean distance is meaningless to me because I can't actually go through the apartment building. And the real distance that I have to go to get to this second point is I have to walk around the building and then down the block again. Okay, so the point being is that none of these distances are the real distance and none of these distances are the wrong distance. They are just different ways, all equally important and all giving you different information about the relative location of one thing to another. And a lot of these algorithms play with the distance to do what they're doing. Okay. So the next slide, I'm going to skip this. The variance stabilization is not that important, and this is not. So one will see the term kernel measurements in a lot of these algorithms. All that means is distance. They're basically using a different kernel is basically at a high level a different distance metric. Okay, and some other terms, distance and similarity, they're both very similar things, and they just is one over the other. Okay, So that's all they mean. So don't be intimidated. Just dissimilarity is 1 over the distance. Okay. So to go to the next slide. So clustering. So the first thing I'm going to talk about in a little more detail is clustering. It's the easiest one to understand and also very common. So clustering is the process of grouping data in such a way that each group, the data within a group is more similar to each other. Okay. Now again, I want to stop here and stress one point. That sounds very intuitive, right? Oh yeah, of course I know what clustering is. How hard is that? How complicated can it be? Just things are similar to each other in one group and they're similar to each other in another group. And those two groups, the cells in both of those groups are more similar to each other than they are to cells in other groups. But the word they're similar, right? It's a totally arbitrary choice of how do you decide similarity? How do you decide what makes a cell more similar to another? Usually you decide it by a distance metric, but we already saw at least 2 distance metrics. There's probably 30 others that are commonly used. So your choice of distance metric to decide how things are scored in a similarity scale is critical to understanding how things are going to cluster. And without knowing that, you're not going to understand what's going on and what your algorithm's giving you. Okay, so clustering puts cells in groups, and the result of a clustering is a set of labels for each cell or each data point. So the labels can be categorical, meaning that, you know, cell 1 is in group A and cell 2 is in group B, or you can actually have probability ones where cell 1 is 60% chance of being in group B or 40% chance of being in group A. Those are more complicated things to deal with. They exist, but not really much in Flow. All right, so Flow basically, a cell gets assigned to a cluster for better or for worse. And the important thing about clustering is it tells you nothing about what those groups mean. And it tells you nothing about what those clusters are. And sort of to go back to my original point, once you know what those clusters are, once you get those clusters, you have to go back to the dot plots and the histograms and everything that these are supposed to be trying to get you away from to understand what the cells in each cluster actually are. So again, just to reiterate that there's no going away from looking at data in dot plots and histograms and whatnot, because it's the only way to actually interpret the data. Okay. So what is clustering? Well, the requirements for a clustering algorithm are some kind of distance metric that you're going to apply, and that has to be defined, and usually the number of clusters that you're looking for. And the output are the classification labels and a measurement that usually describes the clusters. So once you've gotten clusters, you can visualize them in many different ways. So you can put them on a traditional dot plot. Whoops, sorry. Oh. You can put them on a traditional dot plot and sort of color code them manually by the cluster ID, so you can see where they come up and try to interpret them on a dot plot. FCS Express allows you to display different clusters on these fake histograms, where each histogram peak is a different cluster, and it's just telling you different things about the shapes and characteristics, statistical characteristics of each cluster. And it's all in the manual about what characteristics you can look at per cluster. And you can, let me erase these drawings. You can look at them on heat maps. So here you can see that we have a heat map that shows the different clusters and shows the red arithmetic mean of the B red channel in each of the cluster, and the size of the cluster represents the number of events that were assigned to that cluster. And you can also look at clustering, where each cluster you can get a couple of different statistics in sort of a Trivial Pursuit pie slice type thing, where each pie slice is a different statistic. So there's various ways of trying to understand what the clusters mean, but really at the end of the day, most likely going back to your dot plot and looking at the different clusters across the different populations is the best and most critical way of doing that. okay? So there's a lot of clustering methods. You can see, like, you know, whatever, this is just a small subset of the common ones. And here I want to pause and talk about some terminology that you know, is commonly thrown about and dispel a little some myths. You know, and when people come and talk about these, they're going to talk about unsupervised methods, and oh, my method is really great because it's unsupervised and it works it just works by itself, it's fine. And whenever I hear that, I always laugh, because a clustering algorithm, you can let a clustering algorithm or any of these algorithms run unsupervised, just like you can leave a 3-year-old unsupervised. You can totally leave a 3-year-old unsupervised in your house and go to the movies for a couple hours if you tie the 3-year-old to the couch and leave it food and leave it water and turn on the TV and, you know, baby proof the whole room and do this and do that. Then I'm sure pad the walls, make sure you can't jump off the couch. And then after that, you can totally leave that baby unsupervised for a couple hours. Sure, no problem, right? That's the same as these clustering algorithms. You know, by the time you get to use this quote unquote clustering algorithm that's running unsupervised, someone's chosen a distance metric, someone's chosen tuning parameters, someone's chosen a criteria for when the clustering should stop, someone has chosen how the clustering converges, somebody's made 47 decisions that are, most of them, completely arbitrary, with a, you know, in order to get a very specific goal out of it, but that goal is somewhat arbitrary, and how it achieves that goal is also, there's generally different ways that have very slight differences between them, but can impact the results. So, you know, your unsupervised method that you're handed in this talk of some fancy pants mathematician is really actually a very constrained, very supervised, very rigid approach that will get you a very, very particular answer that may or may not be appropriate for your data. Okay, and that's why it's unfortunately important to understand at least the basic principles of how these work, and so you can evaluate whether this quote unquote unsupervised approach is actually useful for you and your experiments. Okay, so I'm going to walk through one of the simplest algorithms just to give you guys a flavor of how these work. They're often presented as these very, very complex mathematical processes with 7 billion equations, but conceptually they're all generally quite straightforward. All right, so Kmeans is one of the most classic algorithms. It was developed in the late 50s, still used a lot today because it's actually quite fast. And scales easily to very large data sets. And basically, in order to do K-means, you specify an expected number of clusters, and K-means will partition the data into that many clusters. And again, this is the important thing about these algorithms, is that if you tell it, find me 12 clusters, K-means will find you 12 clusters, no doubt about it. Now, whether those 12 clusters are biologically meaningful, whether you actually have 12 clusters in your data, or whatnot, the algorithm doesn't care. It just says that you told it to find 12 clusters and it did that. So you got to bear that in mind with these algorithms. Okay, so how does the K-means and most of these algorithms work at a high level? So you start with a random guess of, you know, so in this example, we said, hey, I want you to find 4 different clusters. And so it starts with a random set of 4 different centroids. And centroid, you know, we're only dealing with 2-dimensional clusters. Centroids are just a center, a middle point in n-dimensional space. Okay, but here we only have 2 dimensions, so I could have just said centers. But we're going to start with a random set of centroids. And then we assign each cell in our data set to the cluster with the smallest distance from that cell to the centroid. And then we calculate the arithmetic mean of all the cells in the cluster and repeat. Okay, and I'm going to go through this slowly because it's really straightforward. Okay, so when we start the process, which we started in the top left, we assigned 4 random clusters that randomly appeared over here. okay? And then what happens is we say, okay, now let's assign every cell to the centroid that it's the closest to. So if we do that, we find that all of these cells here are closest to the green centroid that was randomly placed there. And then all of these cells here are closest to the blue centroid. All of the pink cells that I labeled in pink are closest to the pink centroid, and all of the cells in red are closest to the red centroid. Now what we do is we take all these green cells and we calculate a new centroid, the average position of the average of all of these green cells. And it turns out the average is now here. And for the blue cells, we calculate the new average of the blue cells, which is actually over here. And for the pink cells, we calculate the new average of the pink and the new average of the red. And we do that all over again. Okay, so we rinse and repeat. So again, we now calculate we assign every cell to the cluster that it's closest to. And so now we have all these green cells are closest to the green, the blue cells are all closest to the blue. The pink cells now, what is interesting is there's a very few pink cells at the top. I don't know if you can see that on the GoToMeeting, but there's a very few pink cells on the top that are actually closer to the pink cluster than they are to the green cluster. So they're closer to here than they are to here. Okay, and so they got assigned to the pink cluster. And then of course we got a red cluster. And so what happened is those few cells being assigned to the pink cluster pushed, when you calculate the new average position, pushed the pink cluster a little bit up. Okay? And so then we do that again, and it turns out now there's even more cells from that top cluster that are closer to the pink than they are to the green. All right, and the green cells are over here. And so what's happening is that the centroid is being moved for the green towards there, and the pink cluster is being pushed up towards the top. And so again, you repeat it and you say, oh, now look, there's even more of this cluster. And now, in fact, there's very few cells in the bottom cluster, in the bottom cluster of cells that are closer to the pink centroid. Almost all of the bottom cells are now closer to the red centroid. And so basically the pink centroid is being drawn up more and more. The green centroid is being drawn to the right. And then eventually, if you repeat that, you get that the centroids moved to the centers of those four clusters. All right, and that's the whole process. That's it. You know, there's a lot of computer programming that goes to make that happen efficiently and quickly and do the calculations as fast as possible, but that is basically the whole operation. Okay, so if anyone has any questions on that, please don't hesitate to ask. Okay, and that is a very similar process in almost all of the clustering. So Kmeans is great, it's fast, it's easy. I asked it to find 4 clusters and it found 4 clusters. The only issue is again, we then go back to the details of the algorithm. So it found 4 clusters, but remember, when I said find the average and find the distance, generally Kmeans, if you don't specify anything else, is talking about a Euclidean distance, and Euclidean distances are spherical. And so it does really great if your data is spherical. It finds all of your clusters very beautifully. But what if your data is Gaussian? So here's 4 populations that look like this. There's one population that looks like that. There's another population that looks like that. There's another population that looked like that, and another population that looked like that. Well, you can see that because it is baked into the K-means algorithm that the populations are basically spherical, it does a terrible job of actually denoting your populations of interest and clustering them appropriately with elliptical data. Okay? So again, the algorithms will always find you clusters. It did find 4 clusters, and this actually, if you ran the K-means, it converges, it mathematically converges to the optimal K-means solution, but that optimal K-means solution is biologically irrelevant for you because your clusters don't deal with that. And so that's one of the challenges of K-means and why it's not very often used on its own. It's often used as an initial guess for clusters and an input to other algorithms which are better for non-perfect spherical data. Okay. So, you know, if we start to move on and we say, what are the more sophisticated algorithms? All we have to do, you'll hear the term expectation maximization algorithms. All right, I know that sounds much more complicated. It's exactly the same as K-means. All right, the only difference between it is that instead of calculating a Euclidean distance and calculating an arithmetic mean and starting with a random set of centroids, what you do is you start with centroids from some other algorithm in general. Instead of a Euclidean distance, you pick a different kind of distance. And instead of calculating the arithmetic mean as the new centroid during your iterative process, you use some other statistic to calculate the position of the centroid. But the overall algorithm is exactly the same, okay? And you can see that there's one class of these expectation maximization algorithms are Gaussian mixture models. And you can see that the Gaussian mixture model performs much better at finding Gaussian populations than K-means did, okay? Of course, Gaussian mixture models would perform more poorly if the data was all spherical. So, you know, it's a real trade-off. So K-means and expectation maximization models are both model-based clustering, and they assume that the clusters have a general shape. And the advantages of these are that they're very fast, and they usually only need a couple of parameters or metrics to define every cluster. The disadvantage of them is they do not perform well if the shape of the clusters is not what you expected it to be. And that they do not perform well also if there's a lot of variation in the number of points in the cluster. So if one cluster has 80 or 90% of your data and all the other clusters have a few percent each, the data is pretty random. The mixture models also do, in general, you have to specify the number of clusters again. And so often then we'll talk about some ways so there are ways to auto-determine the clusters. You'll see how actually basic and simplistic those are, although they sometimes still do work. And again, the key element of this and pretty much every clustering algorithm is they will converge to a solution. They will always converge. They will always give you an answer, but it is not necessarily the best answer for you. Okay, and that's kind of the problem, and that's why they all need a lot of checking, and they all need a lot of going and putting them on other 2D plots to see if they're giving you something useful. Okay, so there are other kinds of clustering, like hierarchical clustering. I'm not going to go through these. They're all just different clusterings. They all just work with different distance metrics, and they're not, they're a topic for another time. But I did want to talk about this. So, you know, here's an algorithm that's been present in the literature, the flow means algorithm. And there's two objectives of the flow means algorithm. One is to overcome the limitation that the clusters are circular, because we know our flow data is not circular in n-dimensional space or not spherical. So the approach that they take is to merge spherical clusters together. And if you merge a bunch of spheres together, you're going to end up with a shape that's not a sphere. So we try to merge them together. And also by merging, it's trying to determine what is the optimal number of clusters to have. So that tries to overcome the limitation that if you ask for 20 clusters, it'll give you 20 clusters. This is really trying to say, okay, I'm going to tell you what is the optimal number of clusters, and I'm going to try and find the shape of those clusters. So cluster merging is very, very old. The particular paper that describes the flow means is using the elbow method, which you can see is a very, very old and very, very basic technique, okay? And so what is the approach of these and all other cluster merging algorithms? Is that you choose a number of clusters that is generally way higher than the number you actually expect. So, you know, if you think that there's maybe 10 different populations in your data, you pick 100 clusters and you tell Kmeans or some other algorithm to find 100 clusters. And there's various ways to determine the number to start with. You know, here's a fancy pants mathematical way, and it's just almost just as valid to just pick a big number and just let it go. Okay? You then use a very fast and simple algorithm to cluster the data into that many over-clustered clusters. And then you merge the clusters one by one until you decide to stop merging. So how do you merge the clusters? In the Kmeans algorithm, you calculate the Mahalanobis distance between every pair of clusters. So if I have, you know, 100 clusters, I've actually got 10,000 or 5,000 distance metrics, cluster to cluster distance metrics. And then for all of those, and you notice here that this paper is using a Mahalanobis distance. Now they have some justification in the paper why they use that and why that's the most appropriate, but really that's completely arbitrary. And there's 7 other distances that could have been used to figure out which clusters were the closest together. Okay, but anyway, you merge the closest pair of clusters. And then you get a new merge cluster. So instead of 100 clusters, I now have 99 clusters. And now that new cluster has a center distance, a center position. And now you go and you recalculate the distance between those 99 clusters to each other and merge the 2 closest again and keep going and keep going and keep going. And the important thing is that every time you merge the clusters, you calculate a metric. And the metric that is being used here is the distance between the closest pair of clusters. So you basically keep merging, and as you're merging, you calculate this metric. And at some point, the idea is there's going to be an elbow, okay? And the number of final clusters, that is the quote unquote correct number of clusters, is somewhere at the elbow. All right, so there's a lot of fancy math that goes on, but conceptually it's a really, really straightforward process. Very, very arbitrary. Why K-means and not hierarchical clustering? I don't know. Why Mahalanobis distance and not some other distance? I don't know. Why this metric for the elbow and not some other metric to calculate the elbow? I don't know. I mean, I do know. My point being is that each one of those steps I could have slotted in 6 other algorithms at each step and mathematically justified each one. And it would have given me slightly or dramatically different results because it's pulling out different information about my data. You also notice that at the elbow itself, there are 2 points that look equally as close to that elbow. Which one do you pick? Is it 5 clusters or 4 clusters? I don't know. You know, that's a totally arbitrary decision. But then what you do is eventually you do get a final result that's here. Okay, so, and again, I don't know why the video is not working. I have to figure that out later. But at the end of the day, we started with 10 clusters. We overclustered the data at 10 clusters. And at the end, the Kmeans algorithm told us that we now have 5 clusters. And you can see that, you know, at least looking at FL1 and FL2, it seems that those did a pretty okay job of identifying different populations within my data, and that, you know, there are different populations in the data. Now, is this top left thing, would you really consider this population different than these green ones that just sort of spread out here more? I don't know. You sort of have to look at other detectors, other channels. Maybe it is more appropriate to have picked actually 4 different populations, and this black one would have gone away and merged with the green one, and they're really the same thing. Again, none of these algorithms are perfect. Okay, there's also density-based clustering. This doesn't use a model. This tries to look at just the overall density profile of the data and cluster things based on closely linked dense regions belong to each other, are closer to each other. So you can see that they, you know, the barrier is not just simply the physical distance between the cells, but it uses areas of low density as a barrier to denote where clusters start and stop. Again, it's cool, it can work, it has advantages and disadvantages. The advantages is the clusters can have any shape or size. You don't really need to know the number of clusters because it just finds things based on dense areas are like valleys here, and then these are like mountains. So it just sort of tries to find the mountains in the data. But again, it's a little bit arbitrary determining where one cluster starts and where one cluster ends. How deep a valley do you have to have between mountains before you consider them two different mountains, as opposed to one mountain with just a little dip on the top? Completely arbitrary choice, and there's 47 different ways of doing it. But they do exist, and they also can work. They're generally slower, much slower than model-based clustering, and also they're difficult to describe. You can't just give a single statistic like a mean or a variance of any one of these clusters because they're all weirdo shapes. Okay, and so I think I'm going to wrap it up there and then get on to maybe do a second part of this for those who want down the road, where I'll talk a little bit about SPADE and other ways to represent these clusters and how SPADE in particular uses these clusters to manipulate them and also represent them in a way that's biologically meaningful. But I feel like this gave a little bit of a background to how clustering algorithms work, some of the concepts like distance and that gets played with in order to cluster the data in different ways, and some of the objectives that people are trying to get at when clustering this data is that, you know, either I have too many dimensions or I have too many cells, and literally the computer is just taking forever to deal with any of it, or I just need to be able to I need it summarized. Like I can't look at 40 different parameters. I don't really believe I have 40 different distinct pieces of information. I really need to track, you know, the 19 cell populations I have. How can I track them? How can I find them? And that's the goal of these. And again, I do want to reiterate that at the end of the day, you do have to understand what your algorithms are doing and what, you know, the choice of distance metric pulls out different information about the data. And so what information is it trying to pull out, whether or not it's succeeding, at least to understand what it's trying to pull out. Is that appropriate for your data? And of course, to always go triple, quadruple check when you're looking at your clusters from a clustering, go back to your dot plots and histograms and confirm to yourself what these clusters are actually pulling out from your data. Okay. So I think that's going to be it for now. Are there any questions? Please feel free to type them in the question and answer section. And please keep an eye out for future webinars that we'll do where I'll elaborate and go forward on this and talk about SPADE and some of the other clustering algorithms in more detail, and this background will come in very useful. Otherwise, for people who don't have any questions and want to leave, I want to thank you very much for attending. I really appreciate it. I hope you found the seminar useful. And if you have any questions about the specifics of anything in FCS Express, of course, write us into support@denovosoftware.com and we will be glad to help.